Hello everyone, welcome to BioWise with Aanchal. Now this video is going to be the second part of the video which I have already made before, that is genetics. Genetics part 1 video is already uploaded. Those who have not seen it, the link is given in the description. Now children, I have tried my best to give you the maximum on this channel. There is a self-assessment form in the description followed by certain notes. Right? So kindly solve those sheets. People are seeing the video but they are not solving the questions. Trust me, if you pay attention, listen to my videos, solve the assessment and also see the notes, it is going to help you. And the charge for this is nothing. You have to pay attention for it. Only that much is needed. And those who have not subscribed the channel, you are requested to subscribe and then proceed so that you get the regular updates. So let us continue with the next topic, that is the continuation of the previous topic, genetics part 2. Okay. In this part we are going to study about Mendelian genetics, that is Gregor Johann Mendel work, and sex linked inheritance. These topics were pending. Now in your exams 100% sure out of the 3 laws, 1 law is going to come. In 2025 law of independent assortment was asked. So law of dominance, law of segregation and even law of independent assortment has to be by heart. It's better that you put these laws in front of your eyes. These are the 3 statements that you need to know for your exams and it is going to be asked for 100%. Okay. So now let us understand Mendelian genetics. First of all, I'll write something for you. Right? I go party. What are these? These are the 7 traits that Mendel has used to carry out his work on the pea plant. Okay. What is I? Inflated pod. This is green pod. This is purple flower. This is axial flower. Right? R is round seed. T is tall plant. And Y is yellow seed. Okay. Now these are the 7 traits that Mendel has studied and these are the dominant traits. So all these you just keep in your mind that none of the alphabet should be repeated. So inflated we use capital I, green we use capital G, purple P, axial A, yellow Y, tall T and round R. So the recessive trait of all these characters will be, for inflated pod you have constricted pod, for green pod you have got yellow pod, for purple flower you have got white flower, for axial flower you have got terminal flower, for yellow seed you have got green seed, for tall plant you have got dwarf plant and for round seed you have got wrinkled seed. So all the dominant traits will be represented by the capital letter of the trait and all the recessive trait will be the lowercase character of the dominant trait. Is that clear? 7 traits are clear. If you remember this, you will remember the 7 dominant traits of Mendel. Right? So let me clear the board. Now my question is that why did Mendel choose the pea plant? Okay. Now Pisum sativum, the scientific name which can be asked in your exam. We have already discussed how to write the scientific name in my previous video. Now why he chose pea plant? Now pea plant had a very short lifespan. Right? At the same time it had many varieties. You just saw 7 of them, 7 varieties which Mendel has studied. Then pea plants are generally self-pollinated. So you can go for artificial pollination or hybridization or cross pollination, that is according to your will, right? And the varieties which are available in the pea plant had generally alternative forms. Like you have seen, tall, dwarf, purple, white, green, yellow, inflated, constricted. So there are two varieties, two forms of the different varieties that Mendel could see. So this makes pea plant a very apt plant to study about genetics, and this is what Mendel did. Now we will study about the different laws of Mendel. Okay. So laws of inheritance that is given by Mendel. So there are 3 laws of inheritance that Mendel gave. The first law was the law of dominance. Now law of dominance states that out of the 2 contrasting forms or alleles, the one which is dominant will express itself and the one which is recessive will remain suppressed or will be expressed only in a homozygous condition. Is that clear? I will give you the complete statement of the law in the description. Kindly go for the same, okay, for the exams. So now how to explain law of dominance? For this you need to know what is a monohybrid cross. What is monohybrid cross? Monohybrid cross is a cross in which only one character is under study. For example, if I take a character like first monohybrid cross mein we take the parents. So let us take a tall plant and a dwarf plant. So here I have taken only one character in study, that is the height of the plant. So this is a monohybrid cross. Parents are generally pure, that is they are homozygous tall and homozygous dwarf. Now when you talk about the gametes formed, since the 2 alleles are same in the homozygous condition, we take only one as a gamete, correct? So this tall plant can form one kind of gamete, that is T, and dwarf plant can also form one type of gamete, that is small t. So when the first generation is formed, the first generation will be the combination of the 2 gametes, that is capital T and small t. So first generation, all the plants will be tall. All the plants will be tall. So when you talk about the phenotype, that is the physical appearance of the plant, all the plants born in the F1 generation are tall. And when you talk about the genotype of the plant, all the plants of the F1 generation are heterozygous. Tall, right? So all the plant, the genotype you can see is capital T, small t. That is the genetic composition, and genotype will be a heterozygous tall plant. So this F1 explains the law of dominance. The F1 generation explains law of dominance. So what is the law of dominance? Out of the 2 contrasting traits, of a character, the one which expresses itself is a dominant trait and the one which remains suppressed is the recessive trait. This is the law of dominance. For the next law, that is law of segregation, we need to find out the F2 generation of the monohybrid cross. According to this law, the 2 factors, they separate themselves into the gamete without blending with each other or without mixing. That is why this law is called the law of purity of gametes. Let me explain you. So what you are going to study now? Law of segregation. The next law, that is law of segregation. What does law of segregation say? Law of segregation. So what does the law of segregation state? It is also called the purity of gametes. Purity of gametes. Okay, remember this, any question can be asked. So now in law of segregation, you talk about the F1 cross F1, that is capital T small t cross capital T small t. Now F1 became the parent here, and the gametes formed. You can see here there are two alleles. So in gametes, the two alleles will go into the gamete without mixing each other. Each allele can pass into the gamete without influencing each other. So there are two gametes formed here and two gametes formed here. Remember the Punnett square that we used to draw? That is for finding out all the possible combinations. So please remember Punnett square, it has to be a square. You remember, you give the male and the female sign here, write the gametes on the top and write the possible combinations inside. These are all the possible combinations of the offsprings. These are not the 4 offsprings. These are the possible combinations of the offsprings that are formed. Now you can see here in the F2, there is, first thing you need to write is the phenotypic ratio. Phenotypic ratio. What is the phenotypic ratio here? Tall, tall, tall and dwarf. So this reappearance of the dwarf character in the F2 generation proves that the traits have not mixed with each other. So now the ratio will be 3:1, where 3 tall plants, 1 dwarf plant. Is that clear? Next one is the genotypic ratio. Genotypic ratio of monohybrid cross. Genotypic ratio of the F1 is what? Nothing, 1, because there is only one type of progeny that is formed in the F1. All are heterozygous tall. But in F2 you have got 3 different types of genotype. 1 is homozygous tall, 2 is heterozygous tall and 1 is dwarf. So the ratio becomes 1:2:1. Okay, 1 homozygous tall, 2 heterozygous tall and 1 dwarf. So this explains the law of segregation. Next law is law of independent assortment. For that we need to know the dihybrid cross. So let me rub the board, then we proceed with the next law. So now next is law of independent assortment. Law of independent assortment. Now in this case we need to know about the dihybrid cross. As seen in the dihybrid cross, during the formation of gametes of the F1, out of the 2 characters, I am talking about 2 different characters. It can be a height or a color of the flower, or a height or the position of the flower, or you can say the color of the flower and the shape of the pod, anything you can take. So in that case, out of the 2 characters, when the separation of the alleles happen, the allele separation into the gametes are independent of each other. You are understanding? I will explain you with the cross. Now in this case, let us take the example of round and yellow seeds. And what is the recessive trait for that? Wrinkled and green seeds. Okay, so this is round and yellow, and this is wrinkled and green. If you have to understand, this is round and yellow, and this is green and wrinkled. This is the parent, and parents are pure, so you can see they are completely dominant, completely recessive alleles are present. Okay, so now when they have the gametes formation, that is the gametes are formed, since they have 2 types of dominant alleles only, the gamete formed is RY, and the going of this Y into the gamete or going of this R into the gamete is independent of each other. Similarly, here you can see the gamete formation will be small r small y. And what will be the F1 formed here? F1 will be capital R, capital Y, small y. Remember, whenever you have to write, the same alphabet has to be written together, capital first, small later. Capital R, capital Y, small y. This is the F1 generation. So what is the phenotype? It will be round, because round is a dominant trait, and yellow, because yellow is also a dominant trait. So here also the kind of seed formed is round and yellow. Is that clear? This shows that round and yellow are the two dominant traits. Recessive ones are not able to express themselves. Okay. So F1 speaks about law of dominance also. Is that clear? So for the next F2 generation and the ratios, I have to clear the board. We have already seen what is the F1 trait. Now we have to talk about the F2. So now F1 cross F1 will give you the F2. So what is the F1? Heterozygous. So these are the two F1s. They are crossed. So gametes formation. So here is a Punnett square to show you the gamete formation of the heterozygous F1. So gametes formed. So there are two things present, that is R and Y. So just write capital R, capital Y, small y. So capital R, capital Y, small r, small y, capital R, small y, and small r, small y. These are the four gametes possible for a F1 variety or F1 progeny. Okay, so this is the Punnett square to show the gamete formation. Now using these gametes, we have to form the possible offsprings. Now this is the Punnett square. You have to write the possible combinations here. So write down the possible combinations along with me. Now pause the video here, draw the grid and write the possible combinations. Then check whether you have written it correctly or not. Then it is going to help. Okay. So let me write it fast. Now these are the possible combinations. So let us see that what are the possible phenotypes. Now wherever you have capital R and wherever you have capital Y, that is going to give you a dominant trait. So let us see, wherever you have capital R, those all things will be round, right? And wherever you have only small r's, that will be wrinkled. Similarly, wherever you have capital Y, even 1, you get a yellow seed, and wherever you have small y, you are going to have green seed. So let us see how many yellows. So round and yellow. Correct? Now in this case, where you have small y, you have round and green. Round and green. Round and green. Where you have small r, you have wrinkled and yellow. So wrinkled and yellow, wrinkled and yellow, and the last one will be green and wrinkled. Is that clear? So you can see here the expressions you can understand well. How many round and yellow are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. So how many round and yellow? You have 9. How many round and green? How many wrinkled and yellow? And how many wrinkled and green? Correct. So how many round and yellow? You have got 9. How many round and green? Round and green, 1, 2, 3. How many wrinkled and yellow? Wrinkled 1, 2, 3. And how many wrinkled and green? 1. So this becomes your phenotypic ratio of the F2 generation for a dihybrid cross. 9:3:3:1. You can see here in the F1 generation also you had the ratio 3:1. And 9:3 is what? If you simplify, it's 3:1 only. So just a way to remember, 9:3:3:1 is your phenotypic ratio. For the genotypic ratio, you need to calculate. In simple, easy way, if I have to tell you the genotypic ratio, let me rub the first line only. For the genotypic ratio, what was the genotypic ratio of the F2? 1:2:1. Write that, double it. 2:4:2. Then write again the same thing. 1:2:1. So this is the genotypic ratio of the F2 generation. In exam generally it is not asked, and it is in your book also, it's written very complex. It's not that complex, if you remember it's easy. 1 completely dominant, 2, then again this one, then this and this, then you have all these 4 as same, and you repeat the mirror image, whether you have the similar structure here. Is that clear? This is your genotypic ratio of the F2 generation. Now here you can see that how all these combinations are possible. It is because this R and this Y, or small r, small y, they are totally independent of each other when it happens in the gamete formation. So this R, this Y, they are going into the gametes independent of each other. This is what the law of independent assortment all about. Any doubt? Put it in the comment section. Okay. Now let us have a very small summary. Okay. Now when you talk about the monohybrid cross, then you talk only about one character, that is tall and dwarf. Correct? You get an F1 and you get the F2. Right? This one is the parent, this one is the F1, and this one is the F2. In F1, we have all the progenies of the same type, but in F2, we have got 3 tall and 1 dwarf, right? But here it is all tall, correct? So here in this case, the phenotypic ratio is 3:1, important, and the genotypic ratio is 1:2:1, again important. Correct? Now when you go to the F2 generation, let us take 2 characters what we have taken, that is round and yellow. It was crossed with wrinkled and green. These were the parents. Here in this case, F1 was round and yellow, but it was heterozygous, and F2 round and yellow, that was 9 out of 16. Then you had wrinkled and yellow, that was 3 out of 16. Then you had round and green, 3 out of 16, and the last me you had wrinkled and green, that is 1 out of 16. Okay, so this was the F2. Fill in the blanks mein you can write capital R, small r, capital Y, small y. So both the characters you can write in uppercase or lowercase. Okay. So what is the F1 ratio? 9:3:3:1. And what is the genotypic ratio? 1:2:1 2:4:2 1:2:1. This is the genotypic ratio. So this was the F1 and the F2. Here you prove the law of dominance, here you prove the law of segregation, and here you have to prove the law of independent assortment. Is that clear? Put it in the comment section if it is not clear. Next topic is sex-linked inheritance. Now, what is sex-linked inheritance? The X chromosome or the Y chromosome which is present in human beings to determine their gender, if any kind of disease is related to these X chromosome and the Y chromosome and can be transferred to the next generation, it is called X-linked disorder or trait. So, a specific trait which is present only on X chromosomes exclusively is called X-linked disorder, and if certain genes are present only on Y chromosome, it is a Y-linked disorder. You must have seen that generally the baldness and hair on the pinna is observed in the male and is transferred from father to son, that is Y-linked disorder. X-linked disorder is like color blindness, hemophilia. Hemophilia, the bleeder's disease, we have already studied about earlier. Bleeder's disease is when the blood fails to clot. So, you must have known by now that Queen Elizabeth family, they have the disease in their family line. Okay. So, what are these X-linked disorders? Examples. So, let us study about sex-linked inheritance. In sex-linked inheritance, there are two types of chromosomes, that is allosomes X and Y. So, if it is X-linked, it is called X-linked disorders, X-linked disorders or diseases. For example, you have color blindness, hemophilia. Right. Then Y-linked traits, example hair on the pinna, certain baldness is also some Y-linked, right. So, we will study about how these things are inherited. Okay, we'll take case wise studies. Now, since it is on chromosomes, we do not write capital and small, we write X chromosome. Okay. So, for color blindness, the normal X chromosome is this, and the chromosome with the disorder can be written as XC or it can be written as X0. For hemophilia, it can be written as XH or it can be written as X0. Okay, this is how it is represented. Here it is not with capital letters and small letters. Here you write the X chromosome, give it a superscript. If there is no superscript, that means it is a healthy X chromosome. Is that clear? So, let us talk about cases now. Let me rub the board. Now, the first case that we are going to take, let us take an example of color blindness. The first case that we are going to say is a normal mother and a colorblind father. Okay. If I say normal, that means normal visioned. I am not talking about anything else. It's normal mother means normal visioned mother and a colorblind father. Now, for male, the problem is they have only one X chromosome. So, if that chromosome is diseased or is not good, then obviously the disease will be seen in the males. Now, your question that is asked in the exam is why males generally suffer from X-linked disorders and not the females. Okay. So, this question will be answered after we do all the crosses. Now, normal mother means X and X chromosome and colorblind father means XCY. Okay. So, let us draw the Punnett square to find out the possible combinations. So, X, C, X, Y, X and X. So, the first always write the healthy one first and then the diseased X, Y, X, X, C and X, Y. Now, you can see here that these females are carrying the faulty chromosome. But since they have double set of X chromosome, they are not going to display the disorder, rather they become the carrier. So, if you see the phenotype all the children of this couple are going to be normal visioned. Okay, so they have got 2 carrier daughters and 2 normal sons. Please note here that none of the children are color blind. None of the children are color blind. Is that clear? None of the children are color blind. These 2 are carrying the disorder, but since they have 2 chromosomes, according to the law of dominance, the one which is healthy will be dominant. So, the daughters will not be color blind, but they will be the carrier of the disease. Is that clear? So, let us take the next case in which a carrier female marries a normal man. So, carrier, that is, you can write mother, and a normal father. This is the next case. So, what is carrier female? She will have XC, and normal father is a normal man, XY. So, what are the possible combinations? XY, that is the father, and mother is XC, right? Now, here you can see XX, XY. Both the children are absolutely normal. Next case, the daughter becomes the carrier and the son becomes color blind, right? So, out of the 2 daughters, one is completely normal daughter, one is a carrier daughter, but both of them are normal visioned. Out of the 2 son, 50%, you can write 50% of the son of this couple can be color blind. And if you talk about all the children, irrespective of the gender, 25% of the children are color blind. Okay, 50% of the son is color blind, 50% of the son is normal. So, this couple, if they have son, which is again a 50% chance, out of that 50% chance also there is 50% chance that the son belonging to this couple will be color blind. Okay, daughters are all normal, normally visioned, but one of the daughter will be a carrier. Is that clear? So, next case we will take as a carrier mother and a color blind father. Okay. So, let me rub the board. So, next case is a carrier mother, I have not rubbed that. So, carrier mother and a color blind father. Color blind father. Okay. So, what is the genotype of a color blind father? XYC. Now, what are the possible combinations? That is a carrier daughter, a normal son, a color blind daughter and a color blind son. Okay. So, what do you get it? You get a carrier daughter, color blind daughter, a color blind son, a color blind son and a normal son. Is that clear? 50% of the children belonging to this couple is going to be color blind. Now, having a color blind daughter, this woman is not going to show the disorder. So she will appear normal to the people because she will have normal vision, right? But when a child is born, and possibly if a daughter is born who is color blind, that makes sure that the mother is a carrier. So any question can be asked where the possible combinations are given to you and you find the genotype of the parent, genotype of the parents are given to you and possible combinations of the children is asked. Yes, the last case will be colorblind mother and a colorblind father. In that case, you write the answer in the comment section. What is the possibility of the children to be colorblind for a parent where the mother is also colorblind and the father is also colorblind? The answer in the comment section. Now, next topic in this chapter is mutation. What is mutation? A sudden change in your gene, in the chromosome number or the structure of chromosome that can change the characteristic of a person is called mutation. Matlab physical appearance or any kind of malformation can happen because of mutation. Now, you have already studied in history about World War II. There was an atom bomb incident in Japan, Hiroshima and Nagasaki. Till now, the people born to that place have some or the other problem related to the DNA and its structure. So there was a change or mutation that has happened in the genetic composition of those individuals, and generation after generation they are having the problems because of that radiations. Okay, there is a disease also called sickle cell anemia, where there is only 1 point mutation, one alphabet you can say is changed in the DNA, and the entire structure of RBC changes from biconcave spherical to sickle shape, which leads to many disorders. Now, there is a very interesting fact about sickle cell anemia. Generally, it is seen that the person who is heterozygous for sickle cell anemia do not get malaria, and that is the reason why this gene is naturally selected and still running in our gene pool. That is all for genetics. The important questions are given in the description. Kindly solve the Google forms, see the notes, refer them, that is made for you people only. And thank you so much. Like, share and subscribe, and happy learning, children.